Hi viewers, welcome back to one more other lecture session from this channel. So in this video session we will be studying regarding biomacromolecules and as well as structure of proteins in detail. So here what are biomacromolecules? Prior to the explanation of biomacromolecules in detail, if we consider the acid soluble pool compounds, so here all the compounds which come under the acid soluble pool, their molecular weight it ranges around 18 to 800 Daltons. All the compounds which come under the acid soluble pool, their molecular weight does not exceed more than 1000 Daltons. Is it clear? So let me write it on the board. Acid soluble pool compounds molecular weight, its molecular weight ranges around 18 to 800 Daltons and it won't exceed more than 1000 Daltons. Then in the same way, if we consider the acid insoluble pool compounds, acid insoluble pool compounds, so here all the compounds which come under the acid insoluble pool, their molecular weight it exceeds more than 1000 Daltons. Molecular weight exceeds more than 1000 Daltons. So here what are the various compounds which come under the acid insoluble pool? Here the various compounds which come under the acid insoluble pool, it includes proteins. Is it clear? Proteins, polysaccharides, nucleic acids, and as well as lipids. So here all these are the various compounds which come under the acid insoluble pool. So here by taking the advantage of molecular weight into consideration, here we can classify or here we can differentiate the specific compounds into two different types, and these include biomacromolecules and the other one is biomicromolecules. Is it clear? Biomacromolecules and the other one is biomicromolecules. So here what are biomacromolecules? Here the specific compounds whose molecular weight exceeds more than 1000 Daltons, here they are termed as what? Biomacromolecules. Molecular weight exceeds more than 1000 Daltons. Then in the same way, if we consider the specific biomicromolecules, biomicromolecules are they are termed as biomolecules simply. So here what are biomicromolecules? Here the specific compounds whose molecular weight does not exceed more than 1000 Daltons. Molecular weight does not exceed more than 1000 Daltons. Is it clear? So here we need to understand one key point. So here all the various compounds which are placed under acid insoluble pool like proteins, polysaccharides and as well as nucleic acid, their molecular weight it exceeds more than 1000 Daltons. But here if we consider the specific lipid molecule, its molecular weight does not exceed more than 1000 Daltons. But even though the specific lipid molecule it is placed under acid insoluble pool. Is it clear? All the various compounds which are placed under acid insoluble pool like proteins, polysaccharides and as well as nucleic acids, their molecular weight it exceeds more than 1000 Daltons. So the reason why here they are placed under acid insoluble pool, or here they are termed as what? Biomacromolecules. But here if we consider the particular lipid molecule, its molecular weight does not exceed more than 1000 Dalton, but even though it is placed under acid insoluble pool. So here why the specific lipid molecule it is placed under acid insoluble pool here? So here the answer to this question is very simple, because the particular lipid molecule, it is the chief component of cell membrane or plasma membrane. So if we consider a particular living organism, it may be a plant or animal. So here both the plants and as well as animals, they possess a specific delimiting structure which is nothing but what? Cell membrane or plasma membrane. So here the cell membrane or plasma membrane, it is composed of phospholipids. So here the particular phospholipids, they are widely distributed all over the surface of the living organism. Is it clear? The particular phospholipids which are a part of or which are a component of the cell membrane or plasma membrane, they are widely distributed all over the surface of the living organism. So the reason why the molecular weight of the lipid molecule, though it won't exceed more than 1000 Dalton, but here they are placed under acid insoluble pool. And in addition to this, if we consider one particular living tissue, so let me write it here. Lipids are not strictly biomacromolecules. And in addition, if we consider one particular living tissue, right? So here the living tissue, it may be a plant tissue or it may be an animal tissue. Plant or animal tissue, right? So here both the plant tissue and as well as animal tissue, it possesses or it contains a specific delimiting structure which is nothing but what? Cell membrane or plasma membrane. So once when we degrade the particular plant tissue or animal tissue, all the phospholipids which are a part of the cell membrane or plasma membrane, they undergo aggregation in order to form a specialized structure which is nothing but what? Vesicle. Is it clear? So let me show it here. What is vesicle? So here it is nothing but what? Vesicle. As we all know, one particular phospholipid molecule, it contains two components and these include hydrophilic head and the other one is hydrophobic tail. So always the hydrophilic head, it remains at the exterior side because the particular hydrophilic head, it is capable to form the favorable interactions with the aqueous environment. So the reason why hydrophilic head, it remains at the exterior side, but whereas the hydrophobic tail, it remains at the interior side. So here all these are nothing but what? The hydrophilic head, right? Hydrophilic head. And here it is nothing but what? The hydrophobic tail. Hydrophobic tail. And in addition, here we can observe one more other row of phospholipids here. Here it is the hydrophilic head. And in addition, here we can observe the hydrophobic tail. So here it is nothing but what? Vesicle. So in general, the specific vesicle, it won't dissolve in water because the particular vesicle, it is composed of or it is made up of phospholipids. Phospholipids are hydrophobic in nature. So the reason why here the particular vesicle, it won't dissolve in water. So here all other compounds which are placed under the acid insoluble pool like proteins, polysaccharides and as well as nucleic acids, they possess or they contain some particular charge. So the reason why here all these compounds, they are capable to dissolve in water. But here if we consider the particular lipids, they won't possess or they won't contain any specific net charge. So the reason why here the specific lipid molecules, they won't dissolve in water. Is it clear? So the reason why here the particular lipids, here they are not strictly biomacromolecules. Is it clear? And here we need to understand one more other key point. So let me arrange the things here. If we consider the acid soluble pool, So here acid soluble pool, it roughly represents the chemical composition of the cytoplasm. Chemical composition of cytoplasm. Chemical composition of cytoplasm. But whereas if we consider the acid insoluble pool, acid insoluble pool. So here the acid insoluble pool, it represents the specific compounds which successfully synthesizes within the cytoplasm and as well as various other subcellular organelles. Is it clear? Acid soluble pool, it roughly represents the chemical composition of the cytoplasm, but whereas the acid insoluble pool, it represents the specific compounds which successfully synthesizes within the cytoplasm and as well as various other subcellular organelles. Is it clear? Compounds which synthesizes in cytoplasm and various others subcellular organelles. Subcellular organelles. Is it clear? So here the chemical composition of the living tissue, it is an association of or it is an aggregation of both the acid soluble pool and as well as acid insoluble pool. Chemical composition. Chemical composition of living tissue. It includes acid soluble pool and as well as acid insoluble pool. Acid insoluble pool. And if we summarize the specific compounds which are most commonly observed in case of a particular living tissue according to their abundance, is it clear? So here water occupies around 70 to 90%. Water occupies around 70 to 90% in one particular living tissue. Then in the same way, if we consider the proteins, they occupy around 10 to 15%. Is it clear? And if we consider carbohydrates, they range around 3%. Nucleic acid, they range around 5 to 7%. Lipids, they range around 2%. And ions, they range around 1%. And this is the chemical composition of one particular living tissue. And then after we will be studying regarding proteins. So here what are proteins? Proteins are the polymers of amino acids. Once when many number of amino acids, once when they undergo aggregation, here it leads to the formation of one specific polypeptide chain or protein. Is it clear? So let me erase the things here. And if we consider proteins, so here as I have already mentioned you, proteins are the polymers of amino acids. Proteins are the polymers of amino acids. So here once when many number of amino acids, once when they are capable to undergo aggregation, here it leads to the formation of what? Here it leads to the formation of one specific polypeptide chain or protein. So in general, in order to form or here in order to biosynthesize one specific biological protein, 20 amino acids, they play an important role. Is it clear? So here these 20 amino acids, they are termed as what? Standard amino acids. 20 standard amino acids. 20 standard amino acids participate in the biosynthesis of protein. In the biosynthesis of protein. So here why these specific amino acids, they are termed as standard amino acids? Because all these 20 amino acids they are observed nearly in case of all the biological protein in case of the various living organisms. In case of all the living organisms, these 20 amino acids they play an important role in the biosynthesis of biological protein. So the reason why here they are termed as what? 20 standard amino acid. And one specific amino acid, it is capable to remain in association with the neighboring amino acid by taking the advantage of a specific covalent bond, which is nothing but what? Peptide bond. Is it clear? One specific amino acid, it is capable to remain in association with the neighboring amino acid by taking the advantage of a specific covalent bond, which is nothing but what? Peptide bond. So look at here. So here it is nothing but what? The peptide bond. Is it clear? So here it is one of the amino acid and it is the other amino acid. So here the two amino acids they are capable to associate by taking the advantage of a specific covalent bond, which is nothing but what? Peptide bond. So during the formation of peptide bond, here one specific water molecule it successfully releases here. Is it clear? During the formation of peptide bond in between the two successive amino acid, here one specific water molecule it successfully releases during the formation of peptide bond by the means of a specific mechanism, which is nothing but what? Dehydration. So here it is nothing but what? Dehydration. Formation of peptide bond, it is not a spontaneous process, it is a non-spontaneous process. It requires energy. So look at here. So here it is nothing but what? The peptide bond formation, right? Peptide bond formation. And in this way, all these 20 amino acids, they are capable to remain in association in order to form one specific biological protein. So here most of the biological proteins, they are heteropolymeric in nature. Is it clear? So let me write it on the board. Biological proteins are heteropolymeric in nature. Heteropolymeric in nature. So here what is the meaning of heteropolymeric in nature here? If we consider one specific protein, it contains many number of amino acid residues. Is it clear? So let us assume some of the amino acids like lysine, arginine, histidine, phenylalanine, tryptophan, tyrosine, alanine, valine. Is it clear? So here one specific biological protein, it contains many number of different amino acids. Is it clear? Never and ever one specific biological protein, it contains all the similar amino acids. If a specific biological protein, if it contains all the similar amino acids, then here it is termed as what? Homopolymeric. But here most of the biological proteins, they are heteropolymeric. That means one specific biological protein, it contains many number of dissimilar amino acids. Here one specific biological protein, it contains all the 20 amino acids. So the reason why here one specific biological protein, they are heteropolymeric in nature. And in general, the specific amino acids which participate in the biosynthesis of a particular biological protein, generally they are of two different types. And these include essential amino acids and the other one is non-essential amino acids. Is it clear? Amino acids which participate in protein synthesis They are of two different types, and these include essential and non-essential. So at first, if we consider the essential amino acids, so here what are essential amino acids? So here the specific amino acids which cannot be synthesized by our body by the means of metabolic pathways and which are to be obtained through the nutritional diet. So specific amino acids, here they are termed as what? Essential amino acids. Is it clear? So in general there are 9 essential amino acids. So here what are the various 9 essential amino acids here? The various 9 essential amino acids, they include valine, leucine, isoleucine, methionine, threonine, lysine, histidine, phenylalanine, and the other one is tryptophan. So all these 9 amino acids are essential amino acids. These amino acids, they cannot be synthesized by our body by the means of metabolic pathways. They are to be obtained through the nutritional diet. So the reason why here they are termed as what? Essential amino acid. And whereas non-essential amino acids, as the name itself suggests, these specific amino acids, they can be synthesized by our body through the metabolic pathways and they are not to be obtained through the nutritional diet. So the reason why here they are termed as what? Non-essential amino acids. In general, the initiation of protein synthesis takes place at the left-handed side, and here at the left-hand side, N-terminus is present. Is it clear? Initiation of protein synthesis, always it begins only at the left-hand side. At the left-hand side, here we can observe the N-terminus, and at the right-hand side, here we can observe the C-terminus. Initiation of protein synthesis, it takes place at the N-terminus, and at the end of the protein synthesis it takes place at the C-terminus. Is it clear? And here the particular biological protein, it is having many of the biological functions. So here what are the various biological functions of a specific protein here? So let me arrange the things here. And if we consider the biological function of proteins, so here some of the proteins, they act as proteolytic enzymes. So here what are the various proteolytic enzymes here? The various proteolytic enzymes, they include trypsin, chymotrypsin, and as well as carboxypeptidase. So let me write it on the board. Trypsin, chymotrypsin, and as well as carboxypeptidase. So here they are nothing but what? The proteolytic enzymes. Proteolytic enzymes. So here why they are termed as proteolytic enzymes? Because these specific enzymes, they are responsible for cleaving one specific protein at a desirable site. Is it clear? By taking the advantage of proteolytic enzymes, here we can cleave one specific biological protein at the desirable site. So the reason why here they are termed as what? Proteolytic enzymes. They are responsible for inducing the lysis of a specific protein. Then in the same way, if we consider some of the other proteins like antibody. So here what is antibody here? If an infectious agent, if it enters into our body, so here in order to fight against the infectious agent, our immune system, it synthesizes a specific protein which is nothing but what? Antibody. So here the specific antibody, it fights against the infectious agent. Is it clear? And the specific antibody, it is also proteinaceous in nature. Antibody fights against infectious agent. Then in the same way, if we consider the insulin, so here what is insulin here? Insulin, it is the specific hormone which is responsible for converting excess amount of glucose into glycogen, and here the specific insulin, it is termed as peptide hormone. So here why the specific insulin, here it is termed as peptide hormone? Because chemically the specific insulin hormone, it is composed of amino acids. Is it clear? 51 amino acid residues, they are responsible for the formation of insulin. So the reason why here the specific insulin, here it is termed as what? Peptide hormone. Insulin, it is a peptide hormone. And in addition, if we consider some of the transporters like GLUT4. So here GLUT4 is nothing but what? The transporter. So what is the role of the transporter here? As I have already mentioned you, as we all know, the particular cell membrane or plasma membrane, it allows only the hydrophobic molecules to come across it. It won't allow the charged molecules or polar molecules to come across it. So here the specific glucose molecule, it possesses some specific charge, or even it can be dissolvable in water. So here in order to come across the cell membrane or plasma membrane, here the particular glucose, it takes the advantage of a specific transporter, which is nothing but what? Glucose transporter 4. Is it clear? And here the specific glucose transporter 4, it is chemically composed of what? It is chemically composed of amino acids. Is it clear? And in addition, if we consider the specific receptors responsible for binding hormones, as we all know, the endocrine system or the endocrine gland in our body, it synthesizes many of the hormones. So here once when the specific gland, when it synthesizes the specific hormone, here the particular hormone, it successfully transports through the bloodstream, and here the specific hormone, it successfully binds to the target site. So here in order to bind to the target site, here the particular hormone, it takes the advantage of a specific receptor, and here the specific receptor, it is also chemically composed of what? Amino acids. Is it clear? And even if we consider the collagen, so here collagen, it is nothing but what? It is the intracellular ground substance. Is it clear? It is the extracellular matrix protein which is most commonly observed only in case of vertebrates. So here in case of the animal kingdom or in case of the animal world, collagen, it is the most abundant enzyme or protein. Then in the same way, in case of the plant world, rubisco, it is the specific enzyme which is most widely available. Rubisco, it is the specific enzyme which is most abundantly available in case of the plant kingdom. And then after we will be studying regarding polysaccharides. So here what are polysaccharides? So let me arrange the things here. Then after we will be studying regarding polysaccharides. So here what are polysaccharides? So here the acid insoluble pool, it also contains the polysaccharides. So here prior to the explanation of polysaccharides in detail, in general, sugars or carbohydrates, they can be classified into three different types, and these include monosaccharide, disaccharide, and the other one is polysaccharide. Is it clear? Basically sugars or carbohydrates, they can be classified into three different types simply basing on the number of sugar units, and these include monosaccharide, disaccharide, and the other one is polysaccharide. So let me write it here. Sugars or carbohydrates, they are of three different types, and these include monosaccharide, and the other one is disaccharide, and the other one is polysaccharide. Polysaccharide. So here monosaccharide, as the name itself suggests, so here the particular monosaccharide, it contains only one single sugar unit. Is it clear? So here the best examples for monosaccharides, they include glucose and as well as fructose. Glucose and as well as fructose. Then in the same way, the best examples for the disaccharides. Disaccharides, basically they contain two sugar units which are linked by the glycosidic linkage. And the best examples for the disaccharides, they include lactose, sucrose, and even maltose. But whereas in case of the polysaccharide, as the name itself suggests, polysaccharide, they contain many number of sugar units which are linked by the glycosidic linkage. And the best examples for polysaccharides, they are of many types. Basically, the particular polysaccharides, they are of two different types, and these include homopolysaccharide and the other one is heteropolysaccharide. So here, what is the basic difference in between homopolysaccharide and as well as heteropolysaccharide? If a specific polysaccharide, if it contains all the similar sugar units, then here it is termed as what? Homopolysaccharide. Then in the same way, if a specific polysaccharide, if it contains dissimilar sugar units, then here it is termed as what? Heteropolysaccharide. So let me write it here. Polysaccharides, basically they are of two different types, and these include homopolysaccharide and the other one is heteropolysaccharide. So here, what is the basic difference in between homo and hetero? Here homopolysaccharide, they contains all the similar sugar units, but whereas heteropolysaccharide, they contain dissimilar sugar units. The best examples for homopolysaccharides, they include starch, glycogen, cellulose, and as well as chitin. So here all these are the best examples for homopolysaccharides. Then in the same way, the best examples for heteropolysaccharide, it include peptidoglycan and as well as agarose. So here we are not much more concerned regarding the heteropolysaccharide, but here we are much more concerned regarding the homopolysaccharide. So at first, if we consider the starch, so here starch, it is an homopolysaccharide. So let me write it here. Starch, it is an homopolysaccharide. So here the particular starch, which is an homopolysaccharide, basically it contains two specific polymers which are nothing but what? Amylose, and the other one is amylopectin. Is it clear? The particular starch, which is an homopolysaccharide, basically it contains two specific polymers which are nothing but what? Amylose, and the other one is amylopectin. Amylose, which is linked by alpha-1,4 glycosidic linkase. Alpha-1,4 glycosidic linkase. And the other polymer is nothing but what? Amylopectin, which is linked by alpha-1,6 glycosidic linkage. Is it clear? So if you look at the structure of the particular starch, easily we can understand. So here it is nothing but what? The sugar unit, right? All these are the sugar units. Sugar units which are linked by alpha-1,4 glycosidic linkage. So here it is termed as what? Amylose. So as I have already mentioned you, The particular starch, it contains two specific polymers which are nothing but what? Amylose, which is linked by alpha 1,4 glycosidic linkage. So here it is nothing but what? Alpha 1,4 glycosidic linkage. So here why it is termed as alpha 1,4 glycosidic linkage? So prior to that, so here what is alpha configuration and what is beta configuration? If a specific sugar molecule, if it contains hydrogen atom at the top and the hydroxyl group at the bottom, here it is termed as what? Alpha configuration. Is it clear? Here the particular starch, in case of the starch, all the sugar molecules, they are linked by alpha configuration. So here alpha configuration, here it is nothing but what? In case of alpha configuration, hydrogen atom, it remains at the top and the hydroxyl group, here it remains at the bottom. Then in the same way, in case of the beta configuration, hydroxyl group, here it remains at the top, but whereas the hydrogen atom, it remains at the bottom. So here it is termed as what? Beta configuration. So here, as I have already mentioned you, the particular starch, it contains two specific polymers which are nothing but amylose and as well as amylopectin. Amylose, is it clear? Amylose, it is unbranched. Amylose, it is unbranched, but whereas amylopectin, it is branched. So look at here. Amylose, in case of amylose, all the sugar residues, they are linked by alpha 1,4 glycosidic linkage. Is it clear? In case of one sugar molecule, it remains in association with the other sugar molecule by the means of alpha 1,4 glycosidic linkage in case of amylose. But here, if we consider the amylopectin, so let me erase the things here. It is the 6th position, right? So here it is nothing but what? Alpha 1,6 glycosidic linkage. So here most commonly, in case of one particular polysaccharide, reducing end, it remains at the right handed side, but whereas the non-reducing end, it remains at the left handed side. Is it clear? At the right hand side, here we can observe the reducing end, and at the left handed side, here we can observe the non-reducing end. So here the particular starch, it possesses or it contains a specific secondary structure which is nothing but what? Helical structure. Is it clear? So let me erase the things here. The particular starch, it contains a secondary helical structure. Starch contains a secondary helical structure. Is it clear? So here it is nothing but what? The secondary helical structure. To the concerned secondary helical structure, iodine molecule, it successfully binds here. Iodine molecule, right? Helical structure of starch. So to the concerned helical structure, the specific iodine molecule, it successfully binds. As we all know, during the photosynthesis, the specific green plants, they are capable to synthesize the food molecules in the means of sugars. And here, the excess amount of sugar molecules, they remain in the form of what? Starch. So here, in order to prove that during the photosynthesis, green plants, they synthesize the food molecules in the means of sugars. In general, we perform a specific test which is nothing but what? Iodine test. So in case of the iodine test, here we take the advantage of what? Tincture iodine. So here when we pour few drops of tincture iodine on the surface of the particular plant leaf where the photosynthesis has took place. So here the particular part of the plant leaf, it successfully turns into bluish black in color in the presence of tincture iodine. Is it clear? So here the particular iodine molecule, it successfully binds to the specific secondary helical structure of the starch. Is it clear? And in addition, here we need to understand one key point. So here starch is an homopolysaccharide, right? Starch is an homopolysaccharide. Then in the same way, even the cellulose, it is also an homopolysaccharide. But likewise, the particular starch, the specific cellulose molecule, it won't possess or it won't contain the secondary helical structure. Is it clear? The particular starch molecule, it contains secondary helical structure. To the specific secondary helical structure, the specific iodine molecule, it successfully binds. But here, the specific cellulose molecule, though it is an homopolysaccharide, but here the particular cellulose molecule, it won't possess or it won't contain the secondary helical structure. So the reason why here the specific iodine molecule, it is unable to bind to the specific cellulose. Is it clear? Cellulose does not contain helical structure. Is it clear? And some of the other polysaccharides include inulin. Inulin, it is a polymer of fructose. And in addition to this, even in case of plants, there is a requirement of these specific polysaccharides. As we all know, the particular plant cell wall, it is chemically composed of what? Cellulose. So here, in order to acquire the particular cellulose molecule, here the particular plants, they take the advantage of carbohydrate metabolism. In case of plants, the carbohydrate metabolism, it is successfully regulated in case of Golgi apparatus. Golgi apparatus, it is responsible for the biosynthesis of the specific cellulose molecule. And here the particular cellulose molecule, it plays an important role in the biosynthesis of the particular cell wall. Is it clear? So here in case of plants, cellulose is responsible for the formation of cell wall. And in addition to this, here we can observe some more other complex polysaccharides, and these include N-acetyl glucosamine, and the other one is N-acetyl galactosamine. Is it clear? And here the exoskeleton of arthropods. Are in case of the jointed leg organisms. Here the outermost cell. Is it clear? In case of the exoskeleton of arthropods or in case of the jointed leg organisms, here the outermost cell, it is chemically composed of a specific polysaccharide, which is nothing but what? Chitin. Exoskeleton of arthropods contains chitin, which is a polysaccharide. Is it clear? So here this is regarding polysaccharides. Then after we will be studying regarding nucleic acids. So here what are nucleic acids? So let me arrange the things here. And in addition to the polysaccharides, the acid insoluble pool, even it also contains nucleic acids. So here what are nucleic acids? So here basically the particular nucleic acid, it contains many number of heterocyclic carbon rings. Nucleic acids contains many number of heterocyclic carbon rings. So here these heterocyclic carbon rings, here they are termed as what? Nitrogenous bases. So here what are the various nitrogenous bases here? The various nitrogenous bases, they include purines and the other one is pyrimidines. Purines and the other one is pyrimidines. So here the purines include adenine and the other one is guanine, and pyrimidines include thymine, cytosine, and the other one is uracil. So if we look at the structure of these heterocyclic carbon rings, easily we can understand. So here it is nothing but what? Adenine, right? So here it is nothing but what? Adenine. Is it clear? And if we consider the structure of guanine, so here it is nothing but what? The structure of guanine. And if we consider the structure of thymine, so here it is the structure of thymine, right? Thymine. And if we consider the structure of cytosine, so here it is nothing but what? Cytosine. So here all these are nothing but what? The heterocyclic carbon rings, or in other terms here we can consider it as nitrogenous base. So here one particular nitrogenous base, when it remains in association with the ribose sugar, is it clear? One specific nitrogenous base, when it remains in association with ribose sugar, here it is termed as what? Nucleoside. And here to the concerned nucleoside, when the specific phosphate moiety, when it successfully recruits here it leads to the formation of what? Nucleotide. Is it clear? So here it is nothing but what? Nucleotide. Is it clear? Nitrogenous base in association with ribose sugar, here it leads to the formation of nucleoside. And to the concerned nucleoside, once when the specific phosphate moiety, when it successfully recruits here it leads to the formation of what? Nucleotide. So once when many number of nucleotides, once when they aggregate together, here it leads to the formation of what? Nucleic acid. In general, there are two different types of nucleic acids, and these include DNA and as well as RNA. So here these specific nucleic acids, they act as genetic material which is responsible for carrying the genetic information from one generation to the another generation. Is it clear? And then after we will be studying regarding structure of protein. So let me arrange the things here. And then after we will be studying regarding structure of proteins. So here prior to the explanation of structure of proteins in detail, in general, in inorganic chemistry, the structure of compounds most commonly they are elucidated in the means of structural formulae or chemical formulae. So here some of the examples include sodium chloride and magnesium chloride. And likewise in case of the inorganic chemistry, in organic chemistry, the structure of compounds most commonly they are explained in the means of two-dimensional view. And some of the examples include benzene and even the naphthalene. And likewise, the organic chemistry and inorganic chemistry, physicists most commonly they explain the structure of compounds in the three-dimensional view or in case of the 3D structure. And likewise, the physicists, biologists most commonly they explain the structure of proteins under 4 different levels, because the structure of protein it is not so small, it is so vast. So the reason why the structure of proteins most commonly they are explained under 4 different structural levels, and these include primary structure, secondary structure, tertiary structure and quaternary structure. Is it clear? So let me write it on the board. Biologists explain the structure of proteins at 4 different levels. These include primary structure, secondary structure, tertiary structure and quaternary structure. Is it clear? So at first if we consider the primary structure, so here what is primary structure? So in case of primary structure, what exactly happens here? In case of primary structure, all the amino acids they aggregate together here in order to form a specific polypeptide chain. Is it clear? So here all these are nothing but what? The amino acid residues. All the amino acids they aggregate together by taking the advantage of a specific covalent bond, which is nothing but what? Peptide bond. So here once when many number of amino acids once when they aggregate together, here it leads to the formation of what? Linear arrangement of protein. So here it is nothing but what? The primary structure. So here in case of the primary structure, here we can observe the assembly of amino acids by taking the advantage of a specific covalent bond, which is nothing but what? Peptide bond. Then what about the secondary structure? Secondary structure. So here in case of the secondary structure, the specific polypeptide chain it undergoes folding. Is it clear? In case of secondary structure, here we can observe 2 different types, and these include right-handed alpha helix and the other one is beta pleated sheet. So here all these are nothing but what? The atoms, right? Right-handed alpha helix and the other one is beta pleated sheet. So let me write it here. Right-handed alpha helix and the other one is beta pleated sheet. So in general, the right-handed alpha helix, it is stabilized by the means of what? Intramolecular hydrogen bonding. Is it clear? The specific right-handed alpha helix, here it is stabilized by the means of intramolecular hydrogen bonding. And if we consider the particular beta pleated sheet, so here the particular beta pleated sheet, it is stabilized by the means of intermolecular hydrogen bonding. So let me write it here. Right-handed alpha helix stabilized by intramolecular hydrogen bonding. Then in the same way, beta pleated sheet is stabilized by intermolecular hydrogen bonding. intermolecular hydrogen bonding. Is it clear? So here the specific secondary structure, it was first explained by Linus and Pauling. Linus and Pauling. Linus and Pauling, for the very first time, they have explained the secondary structure. And then after we will be studying regarding tertiary structure. So here in case of the tertiary structure, what we observe here. So let me arrange the things here. And then after, if we consider the tertiary structure of protein. So here in case of the tertiary structure of protein, what we observe is, as I have already mentioned you, in case of the secondary structure, here we can observe right-handed alpha helix and as well as beta pleated sheet. So here the right-handed alpha helix, it is stabilized by the means of intramolecular hydrogen bonding, but whereas beta pleated sheet, it is stabilized by the means of intermolecular hydrogen bonding. In comparison with right-handed alpha helix, the particular beta pleated sheet, it is not highly stable. Is it clear? In comparison with the right-handed alpha helix, here the particular beta pleated sheet, it is not highly stable. In order to acquire the stability, the particular beta pleated sheet, it may remain in association with another beta pleated sheet, or even the particular beta pleated sheet, it may remain in association with right-handed alpha helix. So here in order to remain in association with the right-handed alpha helix, here the particular beta pleated sheet, here it takes the advantage of a specialized structure, which is nothing but what? Beta turn. Is it clear? So look at here. So let me show it here. So here it is nothing but what? The beta turn. And here it is nothing but what? The beta pleated sheet. Is it clear? So here the particular beta pleated sheet, it may remain in association with the right-handed alpha helix by taking the advantage of a specialized folding, which is nothing but what? Beta turn. So here the particular beta turn, it is composed of specific amino acids like glycine and as well as proline. Is it clear? Glycine and as well as proline, these are the amino acids which are only observed in case of the beta turn. And in case of normal other protein structures, here we cannot observe glycine and as well as proline, because glycine and as well as proline, they won't have any specific side chain. If in case of one particular polypeptide chain, if glycine and as well as proline are present, under such condition, here bending occurs or here folding occurs. Is it clear? The particular polypeptide chain, it won't remain in linear conformation. Here bending or folding occurs if a specific polypeptide chain, if it is composed of glycine and proline. And most commonly the particular glycine and as well as proline, they are observed only in case of the beta turn. So here by taking the advantage of beta turn, which is composed of glycine and as well as proline, here the particular beta pleated sheet, it remain in association with the right-handed alpha helix. Is it clear? So here likewise, once when many number of beta pleated sheet, when they remain in association with right-handed alpha helix, here it resembles the shape of an woolen ball-like structure. So let me show it here. Woolen ball-like structure, right? Is it clear? So here it resembles the shape of an woolen ball-like structure. So here in order to study the tertiary structure of protein, here we can take the advantage of a specific oxygen binding protein in case of whales, which is nothing but myoglobin. Myoglobin. Myoglobin is responsible for studying the tertiary structure of proteins. Tertiary structure of proteins. Or here the tertiary structure of proteins, they are most commonly observed in case of oxygen binding protein, which is nothing but what? Myoglobin. And most commonly the biological activity of all the proteins they are observed only in case of the terrestrial structure. So here in order to elucidate the biological activity of one specific protein, most commonly the terrestrial structure of protein, it is most commonly preferable. Is it clear? And then after we will be studying regarding quaternary structure of protein. So let me arrange the things here. And if we consider the quaternary structure of protein they are observed only in case of the multimeric proteins. Is it clear? It is observed only in case of multimeric proteins. So here 4 polypeptide chains, they may remain in the form of 2 identical subunits. 4 polypeptide chains, they may remain in the form of 2 identical subunits. 2 identical subunits, right? So here these are nothing but what? The 2 identical subunits. So here one of the specific subunit, it may contain strings of spear-like structure. So here these are nothing but what? The helical structures, right? And because of the presence of the helical structure, here it resembles the shape of strings of spear-like structure. Is it clear? So here it is nothing but what? Beta 2, alpha 2, and here it is nothing but what? Beta 1, alpha 1. And here each of the subunit, in case of the quaternary structure, it may contain the hem prosthetic group. Is it clear? Hem prosthetic group. So here it is nothing but what? The hem prosthetic group, right? So here one of the specific subunit in case of the quaternary structure, it may remain on the surface of the other subunit. So here in order to study the quaternary structure of protein, here we can take the advantage of adult hemoglobin. Is it clear? Adult hemoglobin. Adult hemoglobin, it is the desirable protein in order to study the structure of the quaternary structure of protein. The structure of quaternary structure of protein. So basically the particular hemoglobin, it contains 2 identical subunits, and these include alpha 2 and beta 2 tetramer. Is it clear? Basically the specific hemoglobin, it contains alpha 2 beta 2 tetramer, and here the specific adult hemoglobin, it is composed of 574 amino acid residues. Hemoglobin is composed of 574 amino acid residues. So here 141 into 2 and 146 into 2. So when we add them, here we get 574 amino acids. Is it clear? So here this is regarding the quaternary structure of protein. And as I have already mentioned you, the terrestrial structure of protein, it can be well studied in case of the myoglobin. Here the terrestrial structure of protein, it is stabilized by the means of hydrogen bond and as well as the disulfide bond. Is it clear? So here this is regarding biomacromolecules and as well as structure of proteins in detail. So here I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends. Thank you.